Dear students, today I will deliberate on the topic hybridization in plants. Now, what is meant by hybridization? It is a process by which a cross is made between two plants which differ in at least one set of characters. It's also described as the crossing of two plants or lines of dissimilar genotype. Now, what is meant by a hybrid? So, hybrid is an individual which is produced as a result of the cross between two genetically different plants. So, also it can be described as hybrid is the offspring that has been derived from the parents which differ in at least one set of characters. Now, does this process of hybridization occur without the interference of man in nature? Yes, it does occur. Hybridization also takes place in nature, and that is called as natural hybridization. And in the field of plant breeding, hybridization is brought about, and that process is called as the artificial process. So hybridization can take place either naturally or it can be brought about by artificial process. Now this hybridization is the most effective means of transferring the desirable genes into the cultivated plants from the wild plants of related species or related genera. Now, what is the main aim of hybridization? So, the main aim is to transfer either one or few qualitative characters, for example, disease resistance or improvement of one or few quantitative characters. For example, increase in the number of tillers per plant or increase in the number of grains per spike, or it may be also increase in the weight of the seeds. Now let us look into a little bit about the history of hybridization. Mater in 1716, he was the person who first noticed or recorded the natural hybridization in cotton and corn. Thomas Fairchild in 1717, he produced the first interspecific hybrid, and this was called as Fairchild Mule. And this was a cross between two different species of carnation, that is Dianthus barbatus, commonly called as sweet William. This was crossed with Dianthus caryophyllus. So this is a pic of Thomas Fairchild, and this is the herbarium which he had prepared of the two Dianthus species which he used for the development of the interspecific hybrid. Joseph Colruyter in 1760, the German botanist, for the first time, he used this process of hybridization for crop improvement. And also, he was the first to detect self-incompatibility. Thomas Andrew Knight, he was the person who developed several varieties of apples, pears, peaches, grapes, and currants. Rimpau, a Swedish botanist, he was the first person to develop an intergeneric cross, between breadweed, that is Triticum estivum, and rye, that is Cicale cereal, which later on it gave rise to triticale. So this is a pic of Rimpau, and this is the pic of the triticale along with the two parrots. We shall discuss this in the next few slides. A Russian cytologist by name Judy Karpachenko in 1928, he synthesized an intergeneric hybrid and named it as Raphaino brassica. This was the first instance of a new species, which was applied through polyploid. Now, this is the pic of Judy Karpachenko, and this is a pictorial representation of the cross which he did. So he crossed cabbage, that is Brassica oleracea, with radish, that is Raphanus sativa, and he got the offspring. And about this, we will elaborate a little bit later. Now let us look into the objectives of hybridization. The chief objective of hybridization is to create genetic variation. So let us split this into different objectives. So the first objective is to artificially create a variable population. So what is the necessity of a variable population? Now whenever there is a selection has to be done by the plant breeders, they need to have a variable population to source out any characters which are required. That is why a variable population is required. The second objective is to combine the desired characters into a single individual. The third objective is to exploit and utilize the hybrid varieties. Now, when two genotypically different plants are brought together, then in F1, recombination occurs. And from F2 onwards, because of segregation, many new gene combinations are seen from F2 and the later generation. That is why the F2 and later generations are called as segregating generation. Now, this is what is called as degree of variation. Now, this degree of variation, which is produced in the segregating generations, it, it depends on the number of heterozygous genes which are present in the F1, which in turn is dependent on the number of the heterozygous genes which are present in the parents that were used for this hybridization process. Now let us look into the types of hybridization. So it is of the following types: intra-varietal hybridization. The crosses when they are made between the plants which are belonging to the same variety, they are called as intra-varietal hybridization. Second is intervarietal or interspecific hybridization. So when a cross is made between the plants which are belonging to two different varieties, it is called intervarietal. And because they belong to the two different varieties within the same species, that is why it is called as interspecific hybridization. The typical examples are crossing of two varieties of wheat, that is Triticum aestivum and rice, Oryza sativa, etc. Now the third type is distant hybridization. This in turn is of two types. The first one is interspecific hybridization, also called as intrageneric hybridization. So when the crosses are made between two different species of the plants of the same genus, it is called as interspecific hybridization. And because it involves the same genus, two different plants belong to the same genus, therefore it is called as intrageneric hybridization also. The other type is intergeneric hybridization. So here the crosses are made between the plants which belong to two different genera of the same family. Now both of these, they are called as distant hybridization, and such crosses are called as distant crosses or wide crosses. Now, why is it called as distant crosses or wide crosses? This is because the individuals which are used for hybridization are taxonomically very, or you can say they are more distantly related than the different varieties of the same species. So let us look into the main features of interspecific hybridization. The first feature is it is used when the desirable character is not found within the species of the crop. The second feature is that it is effective when transferring desirable gene into the cultivated plants from their closely related cultivated or wild species. The third feature is it's more common in vegetably propagated species like sugarcane, potato than in seed propagated plants. Now, Feature is it leads to introgression, that is transfer of some genes from one species into the genome of another species. The fifth feature is that as a result of this type of crosses, the offsprings they may be fully fertile, some of them may be either partially fertile or they may be fully sterile in a different group species. So let us look into the characters of the characters or main features of intergeneric hybridization. So the first feature is it is used when the desirable character is not found within the different species belonging to the same genus. The second feature is this method is used only when the specific characters need to be transferred from closely related genera, that is allied genera. The third feature is this method is rarely used in crop improvement by conventional plant breeding, but it can be done, by using tissue culture methods like protoplast fusion and somatic hybridization. This can also be done by a recombinant DNA technology. Fourth feature is this leads to combination of characters of the parental lines. That is, the transfer of some genes from one genus takes place into the genome of another genus. The F1 hybrid, which is obtained over here, is invariably sterile, is always sterile. But the fertility can be restored by doubling of the chromosome by treatment with the colchicine. Now let us discuss few examples of this type of hybridization, interspecific hybridization. So, as we discussed, it's a cross between two plants belonging to two different species, and because it involves two species of the same genus, it's called as intrageneric hybridization. Example is the CO31, which is a superior rice variety. It was developed by interspecific hybridization itself, that is by crossing Oryza sativa variety indica with Oryza perennis, which was drought resistant. The second example is Oryza sativa, which is an Asian upland rice, and it bears characters of high yielding, non shattering nature, and it is resistant to lodging. This was crossed with the African rice, Oryza glaberrima, which was resistant to disease, it was resistant to pests, it was also, it suppressed the weeds, and because of this cross, the rice which was developed was called as Nerica rice. So here's a pig wherein the Asian rice has been crossed with the African rice and we have the Nerica rice. The third example is the Clinton oat variety, which was developed from a cross between Avena sativa and Avena byzantina. The fourth example, the current day cotton shows the um, longer fiber length. So this improved fiber length in cotton. It was developed by a cross between the Indian Gossypium arboreum with the American cultivated Gossypium hirsutum. So here's a pic of Gossypium hirsutum and Gossypium arboreum. Another example Another example I would like to mention is about nobilization of sugar canes. The current day sugar canes which we have, they have got a juicy stem, and in fact we even drink the sugar cane juice. So this was due to the process called as nobilization. So in this process, a cross was made between the noble cane, that is Saccharum officinarum, with the Indian sugar cane, that is Saccharum barbari. Similarly, a cross was made between Saccharum officinarum with Saccharum spontaneum. Now let us look into intergeneric hybridization. So it's a cross between two plants belonging to two different genus of the same family. Now this can develop new crop species. The classical example is of triticale. Now, triticale, which has been obtained, it is of two varieties. One is the hexaploid variety, and the other one is the octoploid. So, let us look into that one by one. So, triticale, it is the first man-made and the only man-made cereal crop. Okay, it has been created by experimental hybridization. So it's an intergeneric cross between wheat and rye. So you can see in the pic over here, the wheat has been crossed with rye and triticale has been obtained. Now let us look into this pic. So cross between wheat, that is triticum durum, which is tetraploid wheat with 2n is equal to 4x is equal to 42. This was crossed with Cicale cereal, which is diploid and it has got 2n is equal to 2x is equal to 14, and the hybrid which was obtained was sterile. It is a triploid where 2n is equal to 3x is equal to 21, so it has got 21 univalents and it is sterile. But then to continue in the F2, what was done was a special method called as embryo rescue method was used, and then this triploid embryo was then uh, treated with colchicine, and this resulted in the doubling of the chromosome to get the hexaploid plant. And since it had the characteristics of both wheat as well as rye, therefore it was christened as triticale, and this is the hexaploid triticale. Now let us look into how the octaploid triticale was developed. So in this, a cross was made between Triticum aestivum, which is a hexaploid variety of wheat, where 2n is equal to 6x is equal to 42. This was crossed with Cicale cereal, that is the diploid rye, wherein 2n is equal to 2x is equal to 14. And the F1 which was obtained, the F1 hybrid was sterile and it was a tetraploid because though it had 28 chromosomes, they were all univalent. Now, this was then subjected to colchicine treatment, because of which there was doubling of the chromosome and an octaploid triticale variety, wherein it was 2n is equal to 8x is equal to 56 was obtained. Now this triticale, it showed improved yield and also in the biochemical components. This triticale was hardy and it can be cultivated even in the arid areas and the semi-arid areas. So arid areas means in places where there is less amount of rainfall and also the soil is less fertile. It can also be grown in wetlands as well as in acid soils. The third example is, let us discuss about wheat, how Triticum dicocoides was developed. So Triticum dicocoides is a tetraploid variety. Now, a cross was made between diploid wheat, that is Triticum boiticum, with a diploid grass by name Aegilops speltoides. So you can look into the genotype. It's A genome in Triticum boiticum, whereas it is D genome in Aegilops speltoides. When a cross was made, and the F1 hybrid which was obtained was sterile because it had got 14 univalents. Now again, by treating it with colchicine and by the process of chromosome doubling, the Triticum dicocoides tetraploid variety of the wheat was obtained. Example 4 is continuing with our story of wheat itself. Let us see how triticum spelta, which is a hexaploid variety, was developed. In 1846, E. S. McFadden and E. R. Sears and H. Kihara independently they artificially synthesized this hexaploid wheat. So they crossed the tetraploid emmer wheat, that is Triticum dicocoides. You can see into the genotype, it is AA and BB, and 2N is equal to 4X is equal to 28. This was crossed with the diploid goat grass, that is Agelops squarosa, and its genome or genotypically it is represented as DT. And of course, it had got 2n is equal to 2x is equal to 14. The F1 hybrid obtained was sterile. You can see it had three genomes A, B, and D. It was a triploid wherein 2n is equal to 3x is equal to 21. That is, there were 21 univalents. So it was sterile. Again, by colchicine treatment, chromosome doubling was brought about, and Triticum spelta, which is the hexaploid variety of wheat, was developed. So you can see the three genomes A, B, and D. So two copies of each genomes are seen. Example fifth one is about Raphanobrassica, which we discussed during the history. So G.D. Karpachenko, a Russian cytologist, he produced this intergeneric hybrid. So what? Just look into this picture. He crossed radish, okay, Raphana sativa with radish, Raphana sativa with cabbage, that is Brassica oleracea, and then he obtained a fertile hybrid, and this was called as Raphano Brassica. Now, just look into this pic, Raphanus sativa, wherein it is having 2n is equal to 18, and the genotype is capital A, capital A. This was crossed with Brassica oleracea. It had same chromosome number, 2n is equal to 18 and BB. So the F1 hybrid which was obtained was sterile. It had A and B genomes, and it had 18 univalents. Now this was then subjected to chromosome doubling by colchicine, and finally the fertile form was obtained, and that was called as Raphanobrassica. In those days, radish was used more as a fodder crop than as a vegetable. You're aware that the root of radish, it stores the food and it is edible. It's the spindle shaped underground part of the root, where the root is modified. The taproot, it's a taproot modification for storage of the food. But radish was relished more as a forage, that is, as a fodder crop for the animals. And it has very fast growth. So, and cabbage, as you know, the vegetative bud of cabbage is edible. So the idea was to combine the desirable characteristic of both of these plants so as to have the the vegetative bud of cabbage on top of the soil and have the roots of radish below. But you can see here in the pic, though Raphana brassica, as per the plant breeding, it was a success, but it did not have utility value because it had the leaves of radish and the roots of cabbage. Now let us look into the differences between interspecific and intergeneric hybridization. So what are the parents involved in the interspecific hybridization? It involves two different species of the same genus. And in intergeneric hybridization, it involves two different genera of the same family. If you look into the fertility, Now these hybrids, they vary. They show a lot of variation. Some of them are completely fertile, some of them are partially fertile, whereas some of them are completely sterile. Now, in case of intergeneric hybridization, hybrids are always invariably sterile. What about the seed setting? Seed setting is more than the intergeneric crosses. And here we can say seed setting is lesser as compared to interspecific crosses. What about its use in crop improvement? This is used quite many times. It is used more than the intergeneric crosses. This one, intergeneric hybridization, is rarely used in the conventional plant breeding methods, but of course it can be used by tissue culture method, wherein the protoplast fusion and somatic hybridization can be done, or even by means of the rDNA technology. Now, what about the release of hybrid variety? I- Uh, so, in interspecific hybridization, it's possible to release the hybrid variety in some crops, but here it's not possible. What about the evolution? Can new crops be evolved? So, in interspecific hybridization, it's not possible, but evolution of new species may sometimes take place. Sometimes it is possible, for example, in example critical in intergeneric hybridization. Now, I have just added this pic. Uh, so this is of Kajanus kajan, that is which we call it as red gram. So these are the two parents, Kajanus platycarpus and kajan, which are taken for the hybridization. It's an example of intraspecific hybridization. So these are two parents, and then you can see this is the F1, okay, which is a tetraploid. You can see the F2, how it appears. Then see the F3, and because the F1 which is obtained, it will be sterile. So the embryo culture has been done, and you can see the hybrid embryo culture, and then multiple shoots arise, and then you get the F1. I've taken this example just to give you a glimpse of what is happening in the plant breeding field. So I showed you about Kajanus in the previous slide. So you can see this is Kajanus plant, the pod, the pod of that plant which has been taken as a female. Well, you can see this is a pod of the Cajanus, which has been taken as a female parent, whereas this one, it is the pod of the Cajanus itself, which is taken as a male parent. And you can see the pod size and the nature of the pod in F1. Okay, and this is in F2, and this is in case of F3. And these are the Kajanasaran, the red gram of the parents, Kajanas kajan and Kajanas platycarpus. So here you can see these are this is the red gram of obtained during F2, and this is obtained during F3. Okay, and this is of course of the back cross. So you can see in this one particular slide all these pics. Hmm, it involves up till F3 generation. So I've just taken this pic over here. So this is of ನೀವು ಕಡ್ಲೆಕಾಯಿ Bengal gram ಅಂತ ಹೇಳ್ತಿರಲ್ವಾ. You can see over here very few pods are there, okay, of sizer. But you can see this one which has been obtained by distant hybridization. Can you see the number of pods over here? There are innumerable pods. Now let us look into some other examples, So improving the crop plants for disease and insect resistance. So you can see in case of cotton, what character was transferred, okay, and from which species it was transferred to which species, from which to which species. So in case of cotton, the Jesset resistance and black arm resistance was transferred from Gossypium tomentosum, Gossypium arboreum to Gossypium hirsutum and Gossypium barbadens. Then in case of okra, this is nothing but our bhindi. So here the resistance to YMV virus, it was this character was transferred from Abelmoschus manihot to Emelmoschus esculenta. Then in case of groundnut, okay, resistant to leaf chewing insect. So this character was transferred from Arachis monticola to Arachis hypogaea. In case of wheat, the rust resistance was transferred from, this is a grass, agropyron. From grass, it was transferred to Triticum aestivum, which is the hexaploid variety of wheat. Then, in case of tobacco, resistance to mosaic virus was transferred from Nicotiana repanda to Nicotiana tabacum. Some other examples where improvement in the quality has been listed. So in case of cotton, the length of the fiber as well as the male sterility, this character was transferred from Gossypium turberi, Gossypium raymondi, Gossypium harkensii to Gossypium hirsutum. In case of potato, you can see the starch content and the frost resistance. These characters were transferred from wild species of Solanum acauel to the cultivated species of Solanum tuberosum. In tomato, the improved carotenoid content was transferred from Lycopersicum wild species to Lycopersicon esculentum. In palm, the quality of the oil, the better quality of the oil, this particular character was transferred from wild species of palm to cultivated species. Similarly, in rice, then oat and rye, the quality of the protein, okay, improvement in the quality of the protein, this character was transferred from wild species to cultivated species. Now let us look into some of the achievements. I've just listed out a very few. You will find hundreds and hundreds and hundreds of examples. So hybrid varieties, the interspecific hybrids. In case of upland cotton, we have MCU2, MCU5, Khandwa 1, Khandwa 2, etc. In case of cotton, Varalakshmi, Savitri, DCH32, NHB12, DH7 and DH9 has been developed right here in India. If you ask your parents, maybe they are aware about Varalakshmi. This Varalakshmi cotton was developed um, along with DCH32 was developed by University of Agricultural Sciences, Dharwad. It was Professor Patil. I think so. I have included his name in the mm, history of plant breeding notes, which I have given it to you. Then bhindi, that is okra, the Parbani variety is an example of an interspecific hybrid. Intergeneric hybrids between pearl millet. Pearl millet is our joda anta ಹೇಳ್ತೀವಲ್ವಾ sorghum, that is also called as pearl millet. So cross was made between pearl millet and grass, that is napier grass, and it has become very popular because it is very high yielding and it yields a very good quality of fodder. So it's for forage, it's for a purpose of forage for the grazing animals here. Okay, they have named it as Jayant and Yashwant. Okay, students, that's it for this class of hybridization. Thank you for your kind attention. Thank you.